So what are the core techniques of modern biotechnology? That is, this modern biotechnology actually it involves the genetic engineering and bioprocess engineering. So what do you mean by genetic engineering? That is the technique in which genetic material, that is either it is a DNA or RNA, is chemically altered. So by means of genetic engineering techniques, we can alter the chemical composition of the DNA or an RNA, and it is introduced into the host organisms. So the modified DNA or an RNA is introduced into a host organism to change the phenotype. So we can change the physical expression, or we can change the particular characteristic, right? We can introduce a particular characteristic into the organism, right? So there is the genetic engineering. So it is a technique in which the genetic material is chemically altered and it is introduced into a host organism. So the altered genetic material is introduced into the host organism, right? So that is the genetic engineering. And what do you mean by bioprocess engineering? That is the maintenance of sterile ambience in chemical engineering processes, right? It is the maintenance of sterile, that is microbial free or without any microbial contamination, right? In chemical engineering processes. So what is the role of maintaining a sterile environment? That is for the manufacture of antibiotics or vaccines, right? So the maintenance of sterile ambience in chemical engineering process for growing the desired microbes or a eukaryotic cell in large quantities, that is fermenters, right? We can grow the microorganisms in fermenters for the manufacture of antibiotics, vaccines, and enzymes. So these two, that is genetic engineering and the bioprocess engineering, are the core techniques of modern biotechnology. So modern biotechnology, it includes mainly the genetic engineering and bioprocess engineering. Okay, clear? So what are the basic steps which are involved in genetically modifying an organism? So what we were doing in traditional or in the olden methods, that is whatever the hybrid that is produced will have both the desirable and the undesirable genes, right? But here, by using the modern biotechnological techniques, we can exactly identify the desirable gene and it can be introduced into the host organism, right? So that is the main advantage here. So the first step is the identification of DNA with desirable genes. So that is the first step in the genetic producing and genetically modifying the organism, right? An introduction of the identified DNA into the host. So after identifying the DNA, the next step is to introduce the DNA into the host. In the third step is the maintenance of introduced DNA into the host and transfer of the DNA to its progeny. So next is the transferring of the DNA into the progeny cells, right? So these are the three main steps which are involved in genetically modifying an organism. First step is identification of DNA with the desirable genes. And then second is the introduction of the identified DNA into the host. And then the maintenance of the introduced DNA in the host and transfer of the DNA into its progeny. Okay, so these are the three main steps. So we'll discuss about it. So what is the basic principle? That is, it is the process of joining and inserting a foreign piece of DNA into a host organism to produce new genetic combinations. So as a result, so normally the plasmid will have its own set of sequences, right? So now the foreign DNA is introduced into that plasmid DNA. So now it leads to a new genetic combination, right? So we are introducing the gene that we want to insert, right? So that is, this technique is called the recombinant DNA technology, right? So it is a process of inserting a foreign piece of DNA into a host organism to produce the new genetic combinations, right? So first the recombinant DNA was produced by Stanley, Hohen and Herbert Boyer in 1972. So they first produced the recombinant DNA. So what they did is, that is, they isolated an antibiotic resistance gene. That is a piece of DNA, right? The gene is nothing but a piece of DNA. So they isolated an antibiotic resistance gene from a plasmid of Salmonella typhimurium. So from the plasmid of Salmonella typhimurium, they just isolated a sequence of nucleotides, right, which is responsible for the antibiotic resistance. And it was linked with the plasmid vector. So this piece of DNA is now linked with the plasmid vector and it is transferred into one E. coli. It is like the same process, right? We saw in the picture, it is as foreign DNA. So here the foreign DNA is nothing but the antibiotic resistant gene from the Salmonella typhimurium. So now this foreign DNA is now combined with the another vector DNA. So this is the basic process